I am Dr. Shipra Goyal, Assistant Professor, Department of Zoology, Kanodia PG Mahila Mahavidyalaya, Jaipur. Today's topic is gene therapy. What is gene therapy? It is an approach of treating diseases by either modifying the expressions of an individual gene or correction of abnormal gene. This can be accomplished by replacing a muted gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functioning improperly, introducing a new gene into the body to help fight a disease. If we see a history of a gene therapy, the concepts of gene therapy was first introduced in 1960s. Fredman and Roblin authored a paper in Science titled Gene Therapy for Human Genetic Disease in 1972. A retrovirus vector system was designed that could efficiently insert foreign gene into mammalian chromosomes in 1984. In 1990, 14 September, the first approved gene therapy in the US at National Institute of Health under directions of William French Anderson. 4-year-old Asanti De Silva received treatment for a genetic defect that left her with adenosine deaminase deficient severe combined immunodeficiency, a severe immune system deficiency. Now, in 1992, Dr. Claudia Bordignon performed first procedure of gene therapy using hematopoietic stem cells as vectors to deliver genes intended to correct hereditary diseases. In 1999, death of Jess Gelsinger in a gene therapy experiment. A research team inserted genes into a brain for the first time in 2003. They use liposomes, which, unlike viral vectors, are small enough to cross the blood-brain barrier. In 2006, use of gene therapy was successful to treat 2 adult patients for X-linked chronic granulomatous disease. First gene therapy trial for inherited retinal disease was done in 2007. An 18-year-old male patient in France with beta thalassemia major has been successfully treated in 2010. Medical community accepted that it can cure HIV, as in 2008, Giro Hutter has cured a man from HIV using gene therapy in 2011. And research is still ongoing. A number of diseases that has been treated successfully by gene therapy increases. There are 2 types of gene therapy, somatic gene therapy and germline gene therapy. Somatic cell gene therapy. That means the therapeutic genes, they are transferred into somatic cells. For example, introduction of genes into bone marrow cells, blood cells, skin cells, any of the somatic cell. It will not inherited later generations. At present, all the researchers, they are directed to correct gene defects in the somatic cells. Germline gene therapy. As with the name suggests, therapeutic gene transform into germ cells, that is eggs and sperm. It is heritable and passed on to later generations. For safety, ethical and technical reason, it is not being attempted at present. Now, one way the germline therapy may be done to ensure a man with a genetic disease doesn't pass it to his child. First, doctors, they remove the man's sperm producing cells which contain a defective gene. Healthy gene is added to each cell to replace the defective one. Then the cells, they are put into mouse testes. They mature inside the mouse and start producing healthy human sperm. Now these sperm, once tested, they are used to fertilize a woman's egg in a laboratory dish. Resulting embryos are placed in a woman's womb and she gives birth to a child whose genes are free from the father's disease. There are 2 types of somatic gene therapy, that is ex vivo and in vivo. In vivo means genes are changed in the cells in the sense when the cells are still in the body. We call it as in vivo because the genes, they are transferred inside the cell. Ex vivo, that means cells, they are modified outside the body and then transplanted back in again. And we call it as ex vivo because because here the cells, they are treated outside the body. There are delivery system, that is in ex vivo and in vivo gene therapy, cell based delivery is there. That is, first, the adult stem cells, they are isolated and propagated in the laboratory. In vitro, differentiated stem cells are taken. Therapeutic gene, that is packaged into a delivery vehicle such as a retrovirus, introduced into the cells. Then now these genetically modified cells, they are reintroduced into the patient. Then there may be a direct delivery. Direct delivery, that means the therapeutic gene, they are packaged into a delivery vehicle such as retrovirus and they are injected directly into the patient to a target organ, for example liver. So example of ex vivo gene therapy is, first, gene therapy, that is to correct deficiency of enzyme adenosine deaminase, that is ADA, performed on 4-year-old girl Asanthi De Silva. She was suffering from severe combined immunodeficiency disease that was caused due to defect in gene coding for ADA. This deoxyadenosine accumulate and destroy T lymphocytes. This will disrupt the immunity and suffer from infectious diseases and they die at young age. Now here, the bacterium carrying plasmid with a cloned normal human ADA gene, right? It is that means it is a cloned ADA gene, and we have taken a genetically disabled retrovirus. Now, this cloned ADA gene is incorporated into a virus, this retrovirus. The T cells with disabled ADA gene isolated from SCI patient, and this retrovirus infect T cells, transfers ADA genes to the cells. Now these cells, they are grown in culture to ensure ADA gene is active, and they are genetically altered cells, they are reimplanted to produce ADA. In vivo gene therapy, it can be only possible option in patients where individual cells cannot be cultured in vitro in sufficient numbers. For example, brain cells. In vivo gene transfer is necessary when cultured cells cannot be reimplanted in patients effectively. If we see some of the examples in vivo gene therapy in patients with cystic fibrosis, a protein called cystic fibrosis transmembrane regulator is absent due to a gene defect. In absence of CFTR, chloride ions concentrate within the cells and it draws water from surroundings. This leads to the accumulation of sticky mucus in respiratory tract and lungs, and treated by in vivo replacement of defective gene by adenovirus vector. Here it is seen that देखिए, this is outside cell and this is inside cell. Here there is a normal CFTR channel, moves chloride ions to the outside of the cell. These are the chloride ions की normal CFTR channel है, तो वो इस तरह से जो क्लोराइड आयंस हैं वो आउटसाइड द सेल मूव कर रहे हैं इजीली. लेकिन अगर CFTR चैनल अगर म्यूटेंट हो गया, दैट मीन्स डज नॉट मूव chloride ions, causing sticky mucus to build up on the outside of the cell. मतलब यहाँ से क्लोराइड आयंस जो है वो फिर वो पास नहीं हो पा रहे, राइट. Normal human gene cloned in bacterium. Normal gene, that is DNA sequence inserted into harmless virus, that is AAV. Virus is dipped into lung through a thin tube that is inserted in the nose. Now here the virus enters the lung cell nucleus. This is a lung cell and this is a nucleus. Now the normal gene, that is normal gene DNA sequence, it is inserted into lung cell DNA by virus, and thus now the cells now with the normal genes, and they will function normally. Some of the key aspects are therapeutic gene, vector, target cell, routes of administration, animal models. First of all is the therapeutic gene. Monogenic diseases, they result from modifications in a single gene that is occurring in all cells of the body. A healthy gene will be introduced. Hemophilia or cystic fibrosis may be the diseases. The multifactorial diseases may be there, that is due to the interaction in more than one gene, or maybe due to environmental factors, that is infections, diet, lack of exercise. Now, if we see the vectors in the gene therapy, vectors means vectors which are used to transfer the desired gene into a target cell. That means we can say a carrier is required. And such vehicles of gene delivery are known as vectors. So there are two main classes of vectors, viral vectors and non-viral vectors. These vectors, they target the right cell, integrate the gene in the cell, activate the gene, avoid harmful side effects, but no universal vector exists. राइट, मतलब अगर हम कहें कि आइडियल वेक्टर में ये सारी चीज़ें होनी चाहिए, मतलब ये सारी चीज़ें हैं, तो हम उसे कहेंगे कि दैट इज़ अ आइडियल और अ यूनिवर्सल वेक्टर, बट देयर इज नो यूनिवर्सल और आइडियल वेक्टर एग्जिस्ट. Viral vectors. Viruses, they introduce their genetic material into the host cell as a part of their replication cycle. They remove the viral DNA and using the virus as a vehicle to deliver the therapeutic DNA. The viruses used or altered to make them safe, although some risk still exists with gene therapy. Cells with the original genes, viral vector carrying new gene. The vector inserts the new gene into the cell and new gene in the cells along with the original genes. There are so many types of viral vectors, that is number of viruses have been used for human gene therapy, that is maybe retrovirus, adenovirus, adeno-associated virus or herpes simplex virus. Retrovirus vector system, that means a recombinant retrovirus have the ability to integrate into the host genome. It carry a DNA of size less than 3.4 kb and target cell may be dividing. Here, the healthy gene, right? It is a healthy gene, that is then retroviruses, they are taken and these are the unhealthy genes. Now these retroviruses and healthy gene, then DNA and nucleus, that is DNA and nucleus, and the healthy gene is inserted here, right? Then the healthy, then they are injected, and that means by the injection healthy cells can be formed. Lentivirus vector system, a subclass of retrovirus. Viral genome in the form of RNA is reverse transcriptase. When the virus enters the cell to produce DNA, which is then inserted into genome at a random position via viral integrase enzyme. Here the target cells are dividing and non-dividing. Now, he's a patient with MLD or WAS. Here the hematopoietic stem cells are taken. Highly purified high titer LV and cytokinins in the petri dish. Then these HCs with the corrected genes. Now the cell, they release without cryopreservation and intravenous infusion of fresh and transduced cells, that means high dose of cells, or they may be done by the myeloablation, that is immunosuppression adapted to the first myelovation, that is immunosuppression कर देते हैं और immunosuppression के बाद में इसमें क्या करते हैं, इसको inject कर देते हैं this transduced cells. ठीक है. Adenoviral vector system does not integrate into genome and is not replicated during cell division. Humans commonly come in contact with adenovirus. Majority of patients have already developed neutralizing antibodies which can inactivate the virus. And here also the target is non-dividing and dividing cells. Now the gene therapy using adenovirus vector. This is an example. Vector binds with the cell membrane, right? यहाँ पर ये क्या है? There is a modified DNA injected into the vector. Means हम कह सकते हैं कि मतलब वायरल डीएनए है, न्यू वायरल डीएनए है, इसके अंदर न्यू जीन डाला है, दैट मीन्स उसको मॉडिफाई किया, इसको फिर एक वेक्टर के अंदर इंजेक्ट कर दिया. अब ये वेक्टर जो है वो सेल मेम्ब्रेन से बाइंड करता है. वेक्टर दैट इज पैकेज्ड इन अ वेसिकल. वेसिकल ब्रेक्स डाउन रिलीजिंग वेक्टर. नाउ द वेक्टर जो है वो क्या है? एडिनोवायरस. वेक्टर इंजेक्ट new gene into the nucleus and cell they makes protein using new genes. Adeno-associated virus vector is a human virus, single-stranded, enters host cell, becomes double-stranded and get integrated into chromosome. This is not currently known to cause disease and consequently the virus causes a very mild immune response. Target cell is non-dividing and dividing cell. Herpes simplex virus vector. They have natural tendency to infect particular type of cell. Herpes simplex virus is a human neurotropic virus mostly examined for gene transfer in the nervous system. If we see a comparison of commonly used viral vectors in the gene therapy, then if we say retrovirus, then the gene material is RNA, packaging capacity is 8 kb, chromosome integration, yes, it infects only dividing cell, persistent gene expression. Adenovirus, double-stranded DNA, 30 kb packaging capacity, there is no chromosome integration, efficient short-term gene expression. Adeno-associated virus, that is single-stranded DNA, 5 kb packaging capacity, no chromosome integration, carries small amount of gene material. Lentivirus, this is gene material is RNA, packaging capacity 8 kb, chromosome integration is there. In fact, both dividing and quiescent cells persistent gene expression. Herpes simplex virus, double-stranded DNA having packaging capacity 40 kb, no chromosome integration, strong tropism for neurons. Now, the advantages are target specific type of cells. They target specific type of cell. They are very good at targeting and entering the cell. They can be modified so that they cannot replicate and destroy the cells. But everything has some drawbacks, that is the disadvantages, that they can cause immune responses in the patients, they can carry limited amount of genetic material and therefore some genes may be too big to fit into some viruses. Now, non-viral vector system, pure DNA construct, direct introduction of pure DNA construct into a target tissue. Efficiency of DNA uptake by cells and expression rather low. Consequently, large quantities of DNA have to be injected periodically. DNA molecule conjugates, that is commonly used synthetic conjugates, that is poly-L-lysine. This is bound to a specific target cell receptor. Therapeutic DNA is then made to combine with the conjugate and form a complex. It avoids lysosomal breakdown of DNA. Lipoplexes, lipid DNA complexes. This is DNA construct surrounded by an artificial lipid layer. Most of it gets degraded by lysosomes. Human artificial chromosomes carry a large DNA, that is with one or more therapeutic genes. Then the methods of gene delivery are, first, if we take physical methods, it is gene gun that employs a high pressure delivery system to shoot tissue with gold or tungsten particles that are coated with DNA. Microinjection, process of using a glass micropipette to insert microscopic substances into single living cell. This is a pipette and this is a cell. Normally performed under a specialized optical microscope setup called a micromanipulator. Then chemical methods, that is using detergent mixtures. Certain charged chemical compounds like calcium phosphates are mixed with the functional cDNA of desired function, and the mixture is introduced nearly the vicinity of recipient cells. The chemicals disturb the cell membrane, widens the pore size, and allows cDNA to pass through the cell. Lipofaction. It is a technique which is used to inject genetic material into a cell by means of liposomes. Liposomes are actually artificial phospholipid vesicles which are used to deliver a variety of molecules including DNA into cells. Now there are many success cases of gene therapy. Let's have a look on it. Gene therapy cures blindness. Blindness of inherited condition. Leber's congenital amaurosis. Inherited disease caused by an abnormality in a gene called RPE65. The condition appears at birth or in the first few months of life, causes progressive worse and loss of vision. How it works actually? It use harmless viruses enable access to the cells beneath the retina of the patients. By using a very fine needle that is safe in an extremely fragile tissue and can improve vision in a condition previously considered wholly untreatable. Then gene therapy reduces Parkinson's disease symptoms. It has significantly improved the weakness of symptoms such as tremors, motor skill problems, and rigidity. It is done with a local anesthesia, used harmless inactive virus AAV. Here, जब Parkinson's disease होती है तो tremors होता है of the hand even at rest, right? और persistent tremors हो सकता है, shuffling gait, unbalanced small steps, curved in a characteristic way, मतलब दिखाई देता है इतना ज़्यादा curved हो सकता है. Advantage of gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, ADA, SCID, etc. It is possible cure for heart diseases, AIDS, cancer. It gives someone born with a genetic disease a chance to life. Right? So it can be used to eradicate the diseases from the future generation. Long disadvantage, long-lasting therapy is not achieved by gene therapy. Due to rapid dividing of cell, benefit of gene therapy is short-lived. Immune response to transfer gene stimulate a potential risk to gene therapy. Disorders caused by defects in multiple genes cannot be treated effectively using gene therapy. Viruses used as a vectors for gene transfer. They may cause toxicity, immune responses, and inflammatory reactions in the host. Ethical issues. Who will have access to the therapy? Is it interfering with the God's plan? Should people be allowed to use gene therapy to enhance basic human rights such as height, intelligence, etc.? Is it alright to use a therapy in prenatal stage of development in babies? So these all are the ethical issues behind gene therapy. So I conclude, theoretically, gene therapy is a permanent solution for genetic disorders, but it has several complexities. At its current state, it is not accessible to most people due to its huge cost. A breakthrough may come any time, and day may come when almost every disease will have a gene therapy. Gene therapy have the potential to revolutionize the practice of medicine. This is a YouTube link or video I am showing, which is also the conclusion of gene therapy. This is a summary of this chapter. These are the references. Thank you for your kind attention.